Welcome to the Banker Lamp. Please enjoy the video.
You will start by confirming that cells need a media change. This saves time and prevents overhandling. First, inspect confluency and morphology under the hood light, looking for contamination or detachment. Next, warm a fresh aliquot of complete media to 37 degrees C and prepare a sterile waste container and pipettes. Once ready, label each vessel with date and passage number so you track conditions. Make sure your gloves and aseptic field are fresh. This step reduces stress and contamination risk. You will now remove spent media while protecting the monolayer. Position the flask or plate inside the biosafety cabinet and tilt slightly. Using a sterile pipette or aspirator, withdraw about 80 to 90% of the media without touching the cell surface. If your protocol calls for a rinse, add pre-warmed PBS gently along the vessel wall and immediately aspirate. This removes residual serum and debris. Once done, avoid letting cells dry. Proceed directly to adding fresh media to maintain osmotic and pH balance. You will finish by replacing with fresh, pre-warmed media and returning cultures to controlled conditions. Slowly add the appropriate volume of complete media down the vessel wall to minimize shear. Once filled, briefly swirl to distribute nutrients evenly. Label and return cultures promptly to the incubator at 37 degrees C, 5% CO2 to stabilize pH. If cells detach or show stress afterward, pause and troubleshoot. Verify wash/aspiration technique, enzyme exposure, and media osmolarity. These are the most common causes of acute detachment.
Thanks for watching. If this was helpful, leave a like and consider subscribing. See you next time.